Marty, thank you. Thank you, everyone, for your time. I'll make this about a 10-minute presentation and then answer questions. I think the Q&A would probably be the most valuable piece. Okay, so we specialize in enriching samples from tissue biopsy for cancer and non-cancer. It increases the purity of cancer cells as they're extracted, and it increases the signal-to-noise ratio for proteomics and genomics and other kind of multi-omic testing. So if I simplify what we do, we create a glass slide, they call it glass. You scan the tissue as opposed to looking under a microscope. You create a negative image, meaning all the things that you don't care about. You create an image for that. You print that mask on top of the actual tissue, and then you extract purified cells and send those off either for sequencing or for mass spec analysis. And then we've completed validation studies with multiple healthcare companies, and we've got a higher VAF signal on genetic testing by 2.7 to 1. And this was the best thing I could come up for a visual for boosted signal. This is 1983 Maxwell high fidelity. So I guess my Google is tuned for the 80s. But that's what we're doing. We're increasing the digital signal. It works for AI, it works for precision medicine, it works for biomarker discovery, it works for cancer-informed therapeutic development, and we use it for all areas of that that you can see here. I'm happy to send a presentation with more of the details. I'll kind of hit the highlights today. Okay, we're working with a little under half of the companies on this list. One company, we're doing biomarker discovery with non-cancer tissue. So we take that tissue, enrich the cells, and then send it off for genetic testing. Another company, we're doing the response to therapeutics. So we'll test in baseline a patient before therapeutic, and then we'll test them again, enriching those cells in both cases to look at the proteomic reaction to therapeutics. We use this with hospitals and companies to get more patients access to life-saving therapeutics. We work with another biopharma company on what we call tissue stewardship. So things like lung cancer have an issue with insufficient tissue. So we boost that signal on the cancer cells because of the enrichment or cellular enrichment. Okay, we're starting a big 4,000 patient study here in New York, and that'll be exciting. That's in collaboration with a biopharma company. Okay, so I think we talked about most of these benefits. Happy to talk more about those later. Would love to answer questions. We did a comparison of kind of the current standard of scraping cancer cells off slides, sending it out for sequencing, and we get better sensitivity and specificity, better precision. We can automate our process, so full automation. So these large testing labs that want to do lights out factory and automate everything, we can do that. And the rest is kind of self-explanatory. Our patents have issued. We filed these patents 5 years ago. And, we talked to our patent attorney. Where's Peter? we talked to our patent attorney, not Peter, different. And they said, oh, it's right around the corner. That was like 4 months ago. And right as we were needing this to be issued, it issued, which is nice. So we estimate that the total addressable market for cancer and non-cancer for our part of the process is probably $4 billion a year. So it's probably big enough to build an operating business. We're working with a fair number of people who we think are good exit partners. So the biopharma companies are probably the best exit partner. The large clinical testing labs are the second best partner, and any of the next-gen health data companies are, as long as they're public, they're a good exit partner from my perspective. Our investors are folks who run those labs. So some of the folks running the labs have invested in the company. Some of the digital AI companies have invested, and we've got healthcare VC money on the deal. From an AI perspective, we use proprietary algorithms to match multiple layers of tissue from stained slides to unstained slides. This is ripe. Everyone at this event has been talking about, where can AI kind of participate in health data or operating businesses. This is an old business, the business of pathology and slides. And we've got a piece where we're digitizing a couple different pieces, automating a couple different pieces, and we're getting better results, and it's prime for AI integration. We've raised $8 million to date. We're closing up on a $2 million funding round. We want that funding done by end of March. So anybody wants to be a participant, just let me know. And then we'll do another round sometime down the road. But all that funding is going to be used for really converting all of the deals that we have in the pipeline right now into larger deals. There's an abundance of customers looking to work with us, and an abundance of customers that are already working with us. Okay, so I think I hit most of these, and I'd love to answer questions. I've got an appendix of other slides that maybe would speak to some of your questions as well. So thank you.
The space thing, why you're in the space, but like this, I mean, you have a lot of business.
Yeah, so a few years ago, I got admitted into a company called Stanford StartX, and it's an incubator out of Stanford University. And it's a pretty special location. They've got 14 unicorns have come out of that. So I was considered one of the CEO founders of one of the companies that they invested in. And in that process, I met John and Badan, who helped run the company with me.
And what led you to decide to work with them? Just because they were part of the incubator, or?
No, it's so this is it's a large industry. It's one of those things where, so there's a lot of, so let me give it a comparison. So if you're in digital pathology, you've got 20 companies doing digital pathology. So that's the process of taking the work of a human pathologist and making it an AI algorithm to automatically diagnose a patient. There might be 20 companies doing scanning. There might be 100 companies doing other kinds of parts of this workflow. But there's really only 3 companies doing any work right now in cellular enrichment, and we compete very nicely against those 2 competitors. So it's got a shot at being a very, very valuable company. Certainly over $1 billion, but it's got a kind of an easy way to get to a couple $100 million. So that's kind of what attracted me to it.
Hey, Chris, Dan Conley. So this is my space. I have a handful of investments, anti-cancer. What you're doing is really cool, as you know. I'm interested in understanding the capitalization, because you just talked about a 2 million safe that's happening now, and then another round to follow soon. So walk us through, because you're going to be hitting free cash flow this year. So there's like 3 or 4 reasons to see that you're on your way. Thank you very much. What was the price of the last round of that, all that 8 million that's been invested so far?
So the round of funding that came in previously was at a higher valuation, call it $20 million or so. The current safe we did was at a 10 million post. And so we sold 20% of the company for $2 million to kind of bridge us to the Series A. The Series A will be roughly, 4 on a 40 or 6 on a 60, something like that. And we won't need the capital except to do an M&A transaction. And so we'd like to hire a good M&A company to help us with that maybe a year from now. We will be cash flow positive this year. We've got 1.6 million of revenue booked, forecasted, approved with just one of the biopharma companies on that list. And so how do I answer your question? So we'll do 2 on 10, and that takes care of all the cash to hit the break even. We'll do 4 on 40 as a bridge to selling the company. And we're already working with several companies who have expressed an interest in buying the technology. An exit on this kind of a deal. So you saw a couple of med tech presentations, and I think the average med tech company will sell, or sorry, the medical device company, like a 510K device, will sell maybe around 350 million or 300 million, something like that. So if we can get a $150 million exit, it's a 10 to 15x for all the current investors.
Well, that's a really thorough answer. Thank you very much.
Sure. Yeah, we thought through it. Yeah.
So just let me understand that you have a technology that a lot of the big guys want.
And they're already working
So
with us.
that's-
Yeah.
Is there a licensing process by which you get some early cash? Sorry. So did I not get that? Now it's filed.
Yeah. So our patents have issued at this point. Certainly licensing is in our future. We've done another batch of patent filings in the last 3 months, and we'll do more patent filings over the spring and the summertime. And licensing is on the list of what we have to do. It would be a good way to capitalize the business, sure.
So right now you're looking for investment banks to help this process, or you're all ready? So
We need investment banks in the summer and fall. Just one investment bank, a good one, that would be great, but we haven't hired one yet. And we need so the current investment round, it looks to be oversubscribed, but we're taking every check that comes in, and it looks like we'll get there. But anybody who wants to invest in the current round, that's still open if people want to. And we haven't set a date for the next round, but it'll be sometime summer or fall.
well, I don't want to hog the mic.
No, it's a good question. So, but it is a special business, and a lot of things have kind of lined up, where the patents have issued, the biopharma companies have signed deals, the large scale clinical testing labs have already worked with us and done projects with us, so revenue generating projects. We have the ability to do so one printer device, one masking device, one part of the workflow from tissue sourcing through sequencing can do 92,000 samples a year. And so it's very high throughput, 400 samples per shift. So everyone wants automation either from liquid handling or throughput or imaging. It's very in line with all of the digital pathology platforms. So we're already partnered with several of those digital pathology companies. One of them has already invested in the company. We're integrated with 3 or 4 of them. Yes. Go ahead. Did you have a question?
I have 1,000 questions.
Sure.
This is fascinating stuff. Let me go back for a second. So do you use any special sauce assays and imaging, any energy, light energy, bioluminescence? What is the modality of energy that really reveals the stuff about the cancer?
Yeah, so we don't do any bioluminescence at all. But think about us as we're a tissue prep process that works well with molecular analytics. So if you want to do proteomics, you can extract proteins from tissue off slides with purified cells. If you want to do genomics and sequencing, you can extract tissue off of slides and send it off for sequencing, and you can do it for very complicated tissue. So this is an example of tissue that is, the subject tissue is spread into a different location. Very hard to remove that off a slide using a manual macro dissection process. And so we enable this kind of tissue analysis. This is another analysis where it's a region containing cell clusters where we're able to get down to less than 50 microns on the analysis of those cells. This is another complicated process where we're able to remove complicated sections of tissue and have purified cells coming off the line in an automated way that is very, very pure in terms of the cells you're extracting. So it's great for research. It's great for testing therapeutics, kind of halfway through a clinical trial for a new biopharma drug. It's great for biomarker discovery. It's got a kind of a world of applications.
Some of the competitors in this space are.
His questions are so good, though. Yeah.
Different from.
Maybe you can explain the difference between this and Thrive.
So for those that know Thrive Bio, they have an amazing imaging ability.
And they've raised a lot of money in our group.
43 million of high net worth money, and 14 million of revenue. So they're off the races. They're in Series B, if anyone is really keen. But I was headed there with the imaging because.
Yeah, so we work with a lot of the imaging companies.
I'm asking him a question first.
Sure, sorry.
Yeah.
So if you can answer the question, then we can get to the discussion on him.
Sure.
So Thrive Bio can look at a stem cell colony, 100 focal planes, microns apart, all the way down, full 3D. And with that, they can watch all the stem cell television of what they respond to and stuff. So
Like they track it with
it's
AI.
similar, but he's going in to grab a slide to find out, well, Thrive is over here growing stem cell colonies. So it's similar, related. I'm worried about the IP on the imaging, but I don't think I should worry. I think I should
Well,
just be appreciative.
let's walk down that lane. Let's walk down that lane since so many people here invested in Thrive.
There could be an overlap.
Well, why don't you ask him?
That's why
Let's discover.
Just met him. It's very fascinating.
We follow.
No, we
that
should,
fast
we
in this
should
place.
talk and compare the two companies. So you're doing 3D imaging of tissue in probably a specialized way. Is that correct?
It's all imaging. They can't touch it with anything because it's a stem cell.
Yeah.
So, look, there's a huge patent estate here. I'm not sure how to answer your question.
Oh, that's okay. But let me just tell you where we come in. So there's an industry where in the US there's probably 20 million cancer tissue biopsies done per year. Worldwide, there might be 100 million tissue biopsies, and we're taking the standard process of glass and tissue and pathology and imaging, and we're jumping into that saying we want to be part of that entire future. And that future includes very high volume testing labs and very, very large pharmaceutical companies that are using it for all the things that I talked about. But it's not 3D imaging. It just isn't. And it's 2D masking for extraction of cellular purification.
So just to address the patent question, because it's what I do. What we do is we look at your patents and we do a landscape analysis of what others are doing to differentiate that, so that we're not investing. Whenever we hear the word overlapping, that's not good. That's a future opportunity for litigation that no one wants, no investors want. So we do a landscape to see what Thrive Bio is doing. We compare it to what you're doing to differentiate it, and we tell the investors it's safe to invest or it's not safe to invest. That's as
It's
straightforward.
not even a matter of being safe to invest. It's a matter of, is this a natural adjacent investment with Thrive so you cover the whole landscape?
Yeah, that's probably the best question, Marty. Yeah.
Hold on a second, we
Just
have
so
another
everyone knows a little bit about Thrive Bio, is that the CEO, founder, Tom Farr-Porsche, he started 18 companies and 7 have become unicorns. So
Yeah, that's unbelievable. Seven unicorns. Truly unbelievable. Yes.
Hi, you report on your website 2 really revenue drivers. That's the cellular enrichment of tissue biopsies, but then you also speak about the QC Precise platform. Is that going to be a large generator for you? And that's the licensing opportunity. Can you elaborate a little on that?
Yeah, so QC Precise is just what our product manager calls the device that prints the masking kind of curable polymer onto tissue. So QuantumCyte, QC Precise, it's all the same thing. And our revenue model is based on recurring revenue. So it's I think you were asking this question, sir, about kit-based revenue and do you have kits? So we do have proprietary reagents as part of our process, and those in the form of kits with that ink and so forth. But it's all based on cellular enrichment, and it's for cancer and non-cancer, and it's for multi-omic testing.
Understood. And then you spoke briefly about Thrive being a competitor. Are there any other competitors of note you can elaborate on?
So there's a small company out of the UK that I'm happy to talk about that's raised about $11 million, but they haven't broken into the market, and we beat them on precision by about 1,000 to 1. And then there's laser capture microdissection, which is equivalent to our precision, but the throughput is way too low to be effective. So we're basically going out there to LCM customers and saying, would you like to use our platform? And they say, yeah, it works well. We can automate our platform and we can get high productivity through our platform. So that's kind of how we win in that scenario. Sure.
Chris, where are you doing all this work? Is it the reason I ask is certain states have life science programs that they make investments. If you look at a state like Texas, they have a $6 billion program for cancer-related technologies. be curious if we can fit that into here, just anything broadly interpreted for cancer research. So
Yeah, I mean, half of our work is with cancer. So, and we definitely fit into that matrix for sure.
what they're looking for on those, and whether it's New York, Texas, or other states, is they're looking for some kind of connection or expansion opportunity to those states to get the money. So we could talk about it separately, but
Sure.
some people have had success with that.
Thank you. I told you guys Q&A was going to be more important than the presentation. So
There's time for one more question if there's from the audience. Or you can just wrap up.
So I'll be around until later today, and fly out at 7 o'clock tonight, and would love to talk to anyone who's interested. Thank you so much.